Yeah. ज्वाइन वो कर नहीं पा रहे हैं ज्वाइन.
मैंने मोदी सर से बात कर दी पैनल मेंबर में ऐड कर
हो गए हो गए.
Good afternoon, everyone. I, Dr. Surbhi Dabral, welcome you all for today's guest lecture, which will be given by our own AIMT alumni of 2013 batch, Dr. Ela Tiwari Jasra, co-inventor de novo cell. We welcome you, ma'am. I would also like to welcome Dr. Amit Chandra Kharakwal, Deputy Director AIMT, and Dr. Jagrati Tyagi, Alumni Coordinator. Now I invite Dr. Jagrati Tyagi on the dais for introduction of Dr. Ela Tiwari Jasra. Over to you, Jagrati.
Thank you, Dr. Surbhi Dabral, and we welcome you, ma'am. Being an alumni, it's very awesome feeling to stand here as I'm also the alumni from the same department. So we welcome you all. So Dr. Ela Tiwari Jasra is an experienced researcher with demonstrated history of working with commercial stem cell industries. She has studied the role of a small molecule in generation of pluripotent cells, stem cells as a source. She is highly skilled and has vast experience in stem cell culture, iPSCs and ESC culture in serum, free and serum containing media, as well as specialized techniques such as making embryonic bodies. She also has advanced knowledge of investigative molecular and cellular techniques such as flow cytometry, RNA analysis, and RT-PCR. She also has extensive experience in working in a GMP grade environment following all GLP and GCP protocols. Strong research professional with a master's degree in medical microbiology from MIT University, Noida, India. Having an insatiable passion for research, she has earned her PhD from University of Malta on derivation of induced pluripotent stem cells from the cord blood. So we welcome you, ma'am. Please come here and join us.
Good afternoon, everyone. Thank you so much for joining. Thank you so much for coming here. And it's a great honor for me to come here and to stand here and to share my research experience and everything which I have gained over past 10 years. And I would like to thank Dr. Amit. I would like to thank the AIMT department for giving me such a strong foundation that I was ready to go out and fight with all the odd circumstances, as well as like professional struggles, everything. So regarding my background, as Dr. Jagrati told you already, like I did my PhD from University of Malta. Then after that, I gained some experience like working in a industrial setup, like where I was working with on the two sides of the industry, like working with the scientific side as well as working with the business side as well. So over the years I have gained experience working with both the industry and academia. So currently, like since past 2 years, I'm working with Alberta Diabetes Institute in University of Alberta at in Canada. And basically this lab is focused on generation of stem cell derived islets for the treatment of type 1 diabetes. So I would like to know, like, in the people who are sitting over here, like how many people like in their families are suffering from this diabetes disease. So I think around the world, and in fact even in US, Canada, and in India also, like the diabetes is a huge issue. Like people who are getting so much of insulin and they are not able to reverse diabetes. They are just pricking all the time to get their insulin tested, like what happens. So then these patients become the no option patient. So when these patient becomes no option patient, then the problem comes, what should be done? Either whole life stay in a insulin, or at the end immunity is highly compromised, people who are suffering from the diabetes. So what is the solution for this problem, how it can be done, and what are the present approaches which people are adopting to fight this disease like diabetes? So right now around the world, like in Canada and even in US, people are going for the islet transplantation. So in islet transplantation, they are taking the help of the human donors, where they are transplanting the islets, and after the transplantation of these islets, people go for the insulin independence for like 8 years, 10 years. But still, it is not a complete solution, because at the end, you are depending upon the donor. Because if there is no donor, you can't get your islet transplantation done. And you are literally limited or restricted with your diet. You are restricted with all, like you have to take so many medications, and which also give your other kind of problems and diseases. So here what I'm going to explain you, like a bit about like what are the advanced approaches to eliminate the off target growth during differentiation of autologous inducible pluripotent stem cell islets in the treatment of diabetes. So so the potential solution these days, what people have started looking for, and there are very few labs around the world who are working in this kind of project, like where they are using stem cells for the treatment of this disease called diabetes. Yeah, sure. So yeah, sure. So right now I'm going to explain you how stem cell derived islets are going to help you for this to overcome this this disease called diabetes, and what are the benefits and what are the disadvantages, and what are tricks and tricks can be adopted to make this happen, like make this world diabetes free or fight against the diabetes. Because I think like everyone who is here, there's one person who is suffering with diabetes in their household or somewhere in their relatives or anywhere else. So let's move forward, and I'm going to explain you like a bit of background, like how it comes and everything. So next slide, please. So this is a brief history, like how it's started, like it was there in 1550 BC, and then there was a scientist known as Benting. He was the person, he discovered insulin, and he was basically his research was basically from University of Toronto. He and his colleague, they discovered the insulin, and they got patent. Like if you see and research about this insulin stuff, they got patent from the University of Toronto. And if you go and see the history about this medicine called insulin, they literally sell off this insulin in $1, because they really want to help the mankind, that I'm not going to take the money out of this patent, because this is really helping patients who are suffering from this disease called diabetes. Can you please move to the next slide? So this is, as I was telling you, the discovery of insulin was done by Benting and his colleagues from University of Toronto, and this is also known as a miracle drug. The reason behind this, it is known as a miracle drug, because people who are suffering from diabetes, literally, they were really in a much more worse condition before this discovery. As you can see in a picture, there was a young girl in 1923, how it was looking like very much thin, and it's very much like about in a state of about to die because of this disease. But after the discovery of this insulin, she was able to recover. As you can see how she is getting healthy. So this is also known as a miracle drug, and it really helped people around the world to overcome this disease. So now next slide. So now the question is, like, anyone can ask, like, insulin is there and people are happy to take insulin, so what is the problem? So the problem is that how far we can go with this insulin therapy. Like all the time you have to use insulin, lifetime, you like till you die, all the time insulin is there. So is it a it is a worthwhile experience, like, or investment, just to depend on the insulin? So this is the huge issue. Like, there are rising insulin prices, like how they are coming up. Prices are there in previously and this year. So the economic burden of diabetes is 1.3 trillion USD, precisely, with no perfect cure of diabetes. So all the time the person has to depend on the insulin for this, because of this insulin therapy. So this is the reason another option came up, that is stem cell derived islets for the treatment of this diabetes. So as the discovery of diabetes is being done by Dr. Benting and his colleagues, so the idea to treat diabetes was like, he did it like, there was two group of patients, healthy patient, and other group was atrophy of exocrine gland. He did it on dogs. One was given like diabetes, and other was also given diabetes, and one was treated with insulin and other was without the insulin. So the dog group which was treated with insulin was completely cured using the insulin. And so this was also so this is the reason this experiment was very much successful, and he demonstrated in animals as well as he demonstrated in humans also. So it was considered as a miracle drug by this scientist, and later on it was very much successful for the people who are suffering from diabetes. So now the question comes, there is also an option for artificial pancreas, there is also an option for islet transplantation. So why islet transplantation, not artificial pancreas? So the major disadvantages of using this artificial pancreas is that you have to depend upon, first of all, on the donor, and that too, after that, the blood glucose levels are not maintained, they are always fluctuating. So this is the major thing which someone has to identify while you are working with this type of disease. And there are two types of cell therapy, like autologous and allogenic. So what's the difference between autologous and allogenic cell therapies? In autologous, your cells are being taken up and they are being processed. They are being processed like minimal manipulation and maximal manipulation. They are being processed, and after that, they are being injected in your own body. So everything which is done on yourself, that is the autologous. And when it comes to the allogenic, in allogenic, patient blood is taken, and after that, the blood is being PBMCs are being isolated, and after isolation of the PBMCs, then they are being distributed into different kind of patient. So this is the reason, and there are disadvantages of using allogenic. The disadvantages of using allogenic is that there is a high risk of forming tumors and cancers, because if they are not compatible, they have the tendency to move towards the cancerous cells. So this is the reason people are moving much feel safer when going to autologous cell therapy as compared to the allogenic one. So why cell and gene therapy are challenging? The cell and gene therapy are challenging because there are different aspect on this, because the starting material is well characterized. In autologous, the starting material has high variability, like you can go for the islet cell therapy, as well as like the product is not personalized. In autologous, like needle to needle traceability is there, so it's lots of issues when it comes to the cell and gene therapy. And it can be so this is the reason people are moving towards the islet transplantation as compared to the just depending upon the donors, so now move towards the next slide. I'm going to show you in the next slide how the workflow takes place. So the workflow for this islet transplantation using stem cells is like, first isolate the qualified donor. After that, cell isolation and cell processing is done. And once the cell isolation and processing is done, then go for the cell expansion. Because in order to make a dose which is equivalent to the human, you need lots and lots of cells. It is not that you can just grow in a petri dish and then you can transplant into a patient, because according to the body mass index, lots and lots of cells are required. And in order to get lots of cells, you need to scale up your process. The scaling up of process is like, you have to use the bioreactors. You have to use large scale materials so that all these stem cells can be scaled up, and you get around 40 million cells, or like more than 200 million cells, just for the one patient. Let's say you have to go for the transplant into 20-30 patients who are enrolled in a study. You need lots of cells. So the companies has moved towards the bioreactors. So I'm going to explain this in the further slide. So why stem cell therapy, and how stem cell therapy can reverse and cure diabetes? As I was telling you, like there is a pancreatic development pathway. Like there is a pluripotent stem cells. Stem cells are there in our like there are two kind of stem cells: embryonic stem cells and adult stem cells. So once you have your pluripotent stem cells, they differentiate into endoderm, pancreatic endoderm, and endocrine progenitor cells. And then finally, they differentiate into beta cells. As you all know, in diabetes and in diabetes dependency or in insulin pathway, beta cell is a very important cell type which is there. So alpha cells, beta cells, pp cells, and these kind of cells which are there. So this is overall which you can see. Oh, change, more. Yeah, this one. So, as I was telling you that this is the pancreatic development pathway. In this pancreatic development pathway, this is the pluripotent stem cells. Pluripotent stem cells. Okay. So, pluripotent stem cells differentiate into endoderm, pancreatic endoderm, and endocrine progenitor cells. And finally, they differentiate into beta cells. And in the beta cells, there are alpha cells, somatostatin cells, pp cells, and gamma cells as well. So this is the reason, like you have, in order to work with stem cells, right from the iPSCs, it can differentiate into beta cells, and there's a different pathway in that whole process. So what I'm trying to do, this is the human development pathway. I'm trying to mimic this pathway in a lab, and then after mimicking this pathway in a lab, once I get the beta cells, I'm going to transplant them into the kidney capsule, or first into the mice, and then I'm going to transplant them into the patients who are suffering from diabetes. The reason behind of this clinical trial is that it gives the patient the insulin dependence for over the years of time. Okay, thank you. So, can you change? Yeah. So, now next slide as well. Next slide, yeah. So, this is just a background, like what is human induced pluripotent stem cells? As we all know that human pluripotent stem cells are being developed by Dr. Shinaya Yamanaka in 2006, and he got a Nobel Prize in 2012. This is a major discovery in the field of cell therapy and stem cell, in the field of stem cell therapy. In the field of stem cell and the regenerative medicine. So, what are iPSCs? iPSCs are formed by introducing the 4 Yamanaka factor as OCT4, SOX, KLF, and CMEC. These are the factors which you can see over here, that is OCT4, SOX, KLF, and CMEC, which are being introduced in adult mice, and after introduction in an adult cell, and after injecting them, these 4 factors into the adult cells using different techniques which are available there, right? Viral factors and other factors, they can able to differentiate them into all the 3 lineages. The 3 lineages are endoderm, mesoderm, and ectoderm. So basically, right now I'm focused on endoderm differentiation, which is like differentiation to beta cells, and beta cells comes under the category of endoderm. And can you click here? Yeah. So these are differentiated into human blood cells derived iPSCs. Next. These are human iPSC cells derived beating heartbeat cells. These are mesoderm. Next. Hope you can see in a picture. These are the red color. These are neuronal cells. These are ectoderm cells. So iPSCs, whenever they are differentiated, they differentiate into 3 types of lineages: endoderm, mesoderm, and ectoderm. So these are the representative images of these, how they are differentiated. Can you change? Yeah. So these are the pioneers. The pictures you are seeing below, like these are the pioneers who are working in this field. Over here, these are, you can see, like Tim Kiefer, Doug Melton, Dr. Kevin, Dr. Christina Nostro, Dr. Mathia Hebrook, Jeff Millman. These are the pioneers who are working in this area of this islet transplantation using stem cell derived islets. And as I told you previously, like pluripotent stem cells, they generate the pancreatic islets using stem cells, and after pancreatic islets are being generated, they are being transplanted into the kidney capsule of a mice in order to know that they are being differentiated in vivo or not, and is there any possibility that using our method we are able to reverse the diabetes into the mice. So next slide. So now how click again, yeah. So now what happens? Like autologous human iPSC islet cell therapy can cure diabetes. Like as I was telling you, the overflow, diabetic patient is there. I collect the blood, and after collection of the blood, I go for the iPSC reprogramming. And iPSC reprogramming is being done using Sandy viruses. And once the iPSC reprogramming is being done, then the PBMCs are being isolated and human iPSC cell line is being generated. And after generation of the iPSC cell line, SC islets are being generated and they are being transplanted into the liver portal islet transplantation is being done into the patients. So what are the benefits of using this autologous cell therapy? So the major benefits of working with autologous stem cell therapy is like unlimited source of supply of cells. You don't have to depend on the donor. You don't have to be onto the waiting list of the donor. Okay, someone is there coming, waiting that, okay, for 1 year, 2 year, then the transplantation is being done. So really don't have to depend on any donor if this therapy comes out to be successful in patient. Right now clinical trials are being done, and there are companies such as Vertex Pharmaceuticals from USA who is already working in this kind of approach, where they have started working with the patient. They have started transplanting these kind of cells into the patient to reverse the diabetes. Recently in 2022, one of the patient is being transplanted with the stem cell derived islets, and he is the first patient who has gone through such type of cell therapy, and till now he has no problems and he is doing well, no diabetes. He is happy to eat everything, sweets, salt, whatever he wants to eat in his in his life. So he's very happy. So if you go and google it, you will always see like Mr. Bryan. He was the first person who is being transplanted from this who got the benefit from this stem cell derived islet cell therapy. And autologous cells will be accepted by the body immune system. So there is no rejection. So HLA typing is being done. There's no possibility of having rejection in this kind of approach. And there is minimal risk of injections and the risk of cancer or mutation, because there's like if we go for the allogenic approach, there's a high risk that the cells can be mutated or they can form cancer. So using this autologous approach, I know this might be a lengthy approach, but this is a safer approach when it comes to the patients. Yeah. So as I was telling you previously, like, okay, you want to transplant into the patients. So you cannot grow the cells in a small petri dish and you can transplant into the patient. That won't be sufficient for anyone. This is just, so what are the possibilities? Like how you are going to do that? So recently, like till now, what is being done around in the lab, around in the world, is that lab scale production of human iPSC islets. So iPSCs are there, they form the islets, and after islets, it is being expanded. Cells are being expanded into the T75 flask. As you can see, this red color flask with blue cap. These are being expanded in that, and they are being transplanted into the mice. So let's say if we are expanding this in flask, how many mice you can transplant? 1, 2, because in mice also you need 2500 IQs of cells to get one transplant done. So it's not sufficient for the mice also. Like after doing like 2 months, 3 months of experiment, you are just transplanting one mice, so that won't give you n is equals to 3. So this is the major limitation of using this petri dishes in this approach. The use of petri dishes is known as a 2D culture. because they are in a planar conditions, and it is known as a 2D culture, and it is not sufficient for the patients. So what is being done now? So the human iPSC islets are being scaled up using the bioreactors. The bioreactors are like the huge reactors which are there, which are being automated, which are being connected to the system. They really like in like 2 weeks or 1 week you can get around 200 million cells. So that really solve this problem of being scale up. Can you move? Yeah. So the islet generation and scale up using 3D bioreactor technology. As I was telling you, the overflow from the diabetic patient, I generate the iPSCs and generation of the iPSCs. Since I'm transplanting into the mice as well as into the patient, it requires lots of quality control. And the quality control are being done using flow cyto, and this is according to the industrial standards. Like this is like more acceptable by the industry as well as more acceptable in academia when you have done this kind of quality control stuff. And so what is being done? Imaging, flow cytometry, genetic analysis, viral screening, and pluripotency gene expression. And then after that, they are being expanded into the flasks, they are expanded into the bioreactors. And this human islet cell differentiation from iPSCs is a 7-stage process. It requires 30 days for this differentiation. If nothing happens, no contamination, nothing, 30 days is being required for this to get this complete differentiation done. So these are the some of the results. As I was telling you, can you click? Yeah. As I was telling you, I reprogrammed this human iPSC cell line using Sandy viruses, day 0 to day 21. As you can see, how the cells arrangement is being changed over the period of time from day 0 to day 21. Like cells were scattered, then after they formed colony, and after at day 21 there was a complete no there was a complete colony shaped structure of iPSCs. By the way, just a quick question over here: how many people know about stem cells and iPSCs? Raise your hand. Anyone heard of stem cells or iPSCs? Sorry. Okay. Yeah, you're right, sir. And anyone from the students? Okay, no worries. It's okay. Okay, no problem. Uh, uh, okay, no problem. So, as I was telling, just you can take an overview, and now you can go and Google it about how things are working, like how things have changed over the period of time, like in past 10 years, like regarding stem cells and iPSCs and where the world is going these days. So, as you can see in the figure B, iPSC colony at day 30, they are very much in a compact size, like how they are, and they have highly tight morphology, and LP staining is being done. Let me change. So this is like a karyotyping analysis and karyotyping analysis of iPSC cells. By the way, karyotyping analysis is being done in microbiology also. Anyone heard about karyotyping here? Okay. So karyotyping analysis is being done in any kind of cells, not about iPSCs or stem cells. It is done in any kind of cells where you have to know you have to know the genetic makeup of cells, how they is there having any chromosomal abnormality or not. Like I have read some papers about, like, say, staphylococcus aureus, where they have done karyotyping analysis to know, like, how what strain can affect and resistant or they are resistant or to different kind of antibiotics. So this kind of test is being done to any kind of cells. So this is a very important test if you really want to publish in a high index journal. Like everyone, they ask for, like, did the karyotyping analysis of these cells, because this gives authenticity that okay, whatever you are giving is to publish, that really means it's not just for to increase the number of the papers. So this is the karyotyping analysis. This karyotyping analysis is being done to know about the chromosomal abnormality. If there is any chromosomal abnormality in any kind of cell, that means it's not going in a right direction. That means something is going on and it has to be looked for. So can you move? Yeah. Okay, just click. So there, as I was telling you, and it is also applicable to microbiology. Like if you are trying to generate any kind of cells like fungus, bacteria, or anything, there are two kind of approaches: 2D or 3D. Like in 2D, they are grown in a petri dishes. In 3D, they are grown in a bioreactors. And the 3D scale up is much more higher as compared to the 2D. So this is, click again. Yeah. So this is like how the cells are there in a bioreactor. Cells are in cluster format. Do you know about this? Anyone knows about this, that in a bioreactor the cells are in a cluster format? Any idea? Okay. So they are in a cluster format in a bioreactor, and it really takes long time for them to develop and to generate. So in order to get this generation done, 150 ml of the media consumption per reactor in 6 days, and the yield is more than 100,000 cells from 20 million cells in 100 ml of a bioreactor. Can you move? This is the immunostaining. I think, who knows about the immunostaining over here? Anyone heard about immunostaining? Okay, so immunostaining is done to know like what kind of markers are there in a once the differentiation is being completed. Like let's say I have generated the iPSC cell line. In the iPSC cell line, there are 4 different kind of markers: OCT, SOX, NANOC, TRA160, TRA181. This immunostaining will give you the preliminary idea. Okay, your cells are good. You can go ahead. You can proceed with your further test. And this is also done using flow cytometry. Just a small advice, since you are upcoming young scientists, that everyone is very important to know about the flow cytometric analysis. And even if you want to work in a field of this microbiology, it's a very important technique, because whatever field you go, it's very important to know the techniques, because techniques are important, which are there and you can apply into any kind of research area. This flow cytometric analysis will give you the idea of like how different kind. Basically it is a quantitative approach, and it gives you the numbers, like okay, this is 56% of your cells are OCT4 positive. Let's say 90% of your cells are nano positive. And then after, you can see in this, can you click? Yeah, this figure you can see, this is the quantitative analysis using flow cytometry, and after that, assessment of teratoma formation using iPSC. So once I got the cell, they are being injected into the mice, and we waited for like 3 months to get the teratoma formation is being done. And after teratoma formation is being done, I went to H&E staining to know like there is a presence of ectoderm, mesoderm, or endoderm or not. So now comes, I was telling you that there are 7 stages of beta cell generation using iPSCs. And what is the problem over here? These are the 2 scientists. Can you click? Yeah, these are the 2 scientists, like Dr. Dieter Ickley and Dr. Lina Sui. They developed a chemical, like I have worked with that, which removes the off-target growth. Whenever we are culturing any kind of cells, let's say in microbiology also, we culture any kind of cells, there is always a possibility of having off-target growth. So in order to remove the off-target growth in cell culture or in a microbiological culture, it's very important to suppress this off-target growth using chemicals, using physical methods. So what these scientists did, like, they have generated phylocolin, and using this phylocolin, up to 70% of C-peptide positive beta cells after 27 days, they got the really pure beta cells in like 30 days of their introduction of this drug to the cells. So can you move forward? So this is like a differentiation in 2D. When I'm saying 2D, that means petri dishes. Everything is being done in petri dishes in 2D. So, and below is, can you click again? Below is the representative images, like how they are being looked when they are generated in a petri dishes. So the total cost in order to generate $2,200 per batch is required to produce 200 IQs from 20 million iPSCs, and which is not sufficient to transplant one mice. So much cost is required, like $2,200 is required. So do you think it is sufficient to transplant into the mice as well as even into the humans? No, it's not sufficient. And you are spending like $2,200 just to get this 200 IQs, which is not sufficient to get the full dose of mice also. So move next. So now next comes, like, there are 7 stages of this IPS differentiation into beta cells. In this 7 stages of this differentiation into beta cells, there are like 4 different markers are being tested. This is the immunostaining results you can see over here, like different like beta cells clusters are there, like PDX1 and K6.1. This is immunostaining is done to know, like, is there any presence of these markers or not. So the 2D culture iPSC derived, so next, 2D culture iPSC derived islets shows the islet hormone but not secreting. So let's say I have developed the iPSC cells. So now what's the question? Like, you have developed the iPSC cell, it should be metabolically and physiologically active also. Otherwise, there's no use of generating this generating this beta cell. It should secrete insulin. And how we can test this in secretion of this insulin is using GSIS. GSIS is an important technique which is there, and it is widely used by many scientists around the world in a very good labs, where they are using this technique to know the level of insulin which is being produced by the cells, which is being produced by the generated stem cell derived islet cells. So you can see over here, can you click again? No, next. Yeah, this one. Okay. So you can see over here, like, there is a secretion of insulin, and which you can see over here at different stages, like static GSIS and dynamic perfusion, which is being done. And there is a process in which cells are being tested into the glucose, and then they are being put into the high level of KCl. And after development of this KCl, then we see, like, what is the level of insulin produced and how actively they are producing insulin. So now next. These are the bioreactors in which in which I have worked, and I have been generating stem cell derived islets in a incubators. These are kept inside the incubators. There is a media inside in the incubator. And just to tell you, like, there are 2 capacity of this bioreactor I have worked. 3 capacities: 100 ml, 500 ml, and 3 liters. And in 100 ml, $4,500 per batch is required just to get beta cells generated, and that can be transplanted into the mice and into the patient. But in the case of 500 ml, $25,000 are being required, and it costs everything. It costs about small molecules, cytokines, whole process, just to know, like, how many beta cells are being generated by the end of 30 days. Move next. Yeah, so this is like a generation of off-target growth. There are ductal cells, ductal tissues which are there. So Next move. So this is like a DNA replication inhibitor which enhances the beta cell maturation, retards the off-target growth. As I was explaining, these are the two scientists, Dr. Dieter Ickley, who was working in this area, and he has developed the drug called filicolin, which affects the pancreatic progenitors of off-target growth and enhance the maturation of cells, and really able to reverse the diabetes. And there was no off-target growth when the cells are being transplanted into the kidney capsule of a mice. Can you please again click? Yeah. So these representative images, which you are seeing, like human iPSC islets, which is there in the middle of it, and human iPSC islets plus filicolin and minus filicolin on the extreme left hand side, you can see they are being compared to the human donor islets. And as you can see in the image, like if I'm not using this drug filicolin, there is an irregular shaped structure which is there in those islets, which is not comparable to the human donor islets. So with the use of this filicolin drug, I got really homogeneous population of these cells, which is comparable to the human donor islets. So this was a huge change which I found with this use of this miracle drug called filicolin, and it really helped to reduce the off-target growth, and really helped it also gives the opportunity to transplant like many mice, and reaching towards the goal of the further clinical trials. Next. As I was telling you that it is a 30 days process of this differentiation from stem cell derived to get this stem cell derived beta cell. And there are different stages. There are like 7 stages, and every stage there is a certain percentage of cell loss. So in the cell loss, which is very much high in stage 2 as compared to the other stages. So maximum amount of cells are being reduced like whenever we are starting of this kind of differentiation. This is the flow cytometric analysis of like stage 1, stage 2, 5 and 7, like how many cells I got at stage 2, 5 and 7, and what is the possibility of going forward, how many beta cells can be required. And this is being done in a real time. Like on the same time I took the cells and did this analysis to know like 80% of these cells are CD117 and CD84 positive. Yeah. So this is like after transplantation into the mice. I did the immunocytochemistry analysis, in which I found that all these cells were positive for insulin in UCN3, GP2, glucagon, C peptide, somatostatin, PDX1. Now move forward. As I was telling you, this is the GSIS analysis for the 3D culture, like the cells which are being generated in a bioreactor. And I found using the filicolin, I found a huge comparable aspect on this. As you can see, the human donor islets are in blue color, and the cells which are treated with filicolin are in green color. So the spike you are seeing over here is very much comparable to the human donor islets. That means that in the lab we were able to generate or I'm not saying in a very ambitious way, but just saying like we tried to mimic the islets which were very much comparable to the human donor islets. So this was a very much encouraging factor for us to move forward, like with experiments and with other aspect of this kind of research. So move forward. Yeah, click more. So this is the patch clamping analysis, and I did this in collaboration with my other team who was there. And in this patch clamping analysis, it is important to know like, is there any secretion of sodium or calcium or not? Because the beta cells which are there in a human body, they have the presence of this there is a presence of this calcium current, as well as this there are sodium channels. So this test is done to know like how much they are comparable to the human donor islets. So next. So this is OCR. I think anyone heard about seahorse and seahorse assay? So seahorse assay is being done. It is also known as the oxygen consumption rate. Like to know, like if you have generated your cells, what is the oxygen consumption rate? Because every cells have mitochondria. So how much percentage of your cells are being active, metabolically active? So this is the reason this test is very important, to know the metabolic and physiological activity of a cells, which is known as the seahorse analysis. This is being done, as you can see in the figure, that they are like the cells which were treated without the filicolin and with the islets, they were very much comparable, and still like human donor islets are found to be very much positive for these kind of cells. Now next, move forward. So this is the gene array. Anyone heard about gene array? So the gene array is being done to know the genetic analysis, like of the cells. Genetic analysis is being done because sometime it happens, even though we have done lots of molecular biology testing beforehand, but still sometime at the genetic level, different kind of mutation or different kind of genetic makeup is being observed in the previous papers. So this is the reason I went towards the gene array analysis, and I did in collaboration with this scientist, Dr. Jickling, in which I found that the cells which I have generated in the lab using bioreactor, they are very much comparable to the human donor islets. And there was a presence, as you can see, the markers, the blue markers over here, which are there, is marked with the blue, that these are found to be in human donor islets as well. So UCN3, insulin, ARX1, these kind of genes are there, which was comparable to the human donor islets. Can you, yeah, again click it again. Yeah. So whatever, as I told you, like I have generated the iPSC stem cell derived islets. So now what next? Okay, it is being generated. The next step is to go for the in vivo analysis. In vivo analysis is being testing into the kidney capsule of the mice. So these clusters are being injected into the kidney capsule of a mice, and then after, blood glucose readings are being taken every 4 weeks till 100 days, to know, like, is there any possibility that they are going to reverse the diabetes or not. And at the same time, blood are being also taken to go for the C peptide analysis using ELISA kit, using an ELISA kit, that to know, like, is there any secretion of C peptide or not, or insulin or not. So by the end of 100 days, I found that some of the mice were able to reverse the diabetes. I did IPGTT. IPGTT is a test which is being done in mice. It is similar to the test which is done in humans. In humans, it is known as OGTT. Like OGTT is being done like whenever someone has to test their sugar levels, like even during the pregnancy, this test is mandatory these days, that by the end of the third trimester, women should go for OGTT analysis. So same way in mice, IPGTTs are being done to take the to know about like what is the actual sugar level in the mice. So now they are transplanted into the mice. After 100 days, I got the graft, and I did the graft analysis. In the graft analysis, I again did this immunocytochemistry, did stain with different markers to know that there is a presence of different markers or not. And luckily, I found everything positive. In the kidney graft, as you can see, there's a separation. This is the kidney capsule, and this is the graft. So there is a presence of all the markers which is required in the stem cell derived in the human donor islets. And on the left hand side, you can see that there is a marginal proliferation of the beta cell. That means that cells which are being transplanted into the mice, they are able to secrete insulin, and they are started developing their, because it takes time in mice to fully mature. So this is the reason it takes time in the mice, like 100 days, even more than 100 days. And this data also gives the safety and efficacy analysis of our study. So now comes the next. So after this immunochemistry, I went again to the flow cytometry, because I want to quantify like how many cells are having actually secreting insulin. And for this quantification, I need flow cytometry. And for this flow cytometry, I did this in a real time. Like I extracted the graph today, and like I did this by the end of the day only, and I found like more than like insulin. There's a presence of insulin. There's a presence of glucagon. There's a presence of C-peptide, as you can see over here, and 50% of the cells were able to produce the insulin, which was a huge encouraging factor for us as a team. So next. So now, what's the whole takeaway of this study? The whole takeaway of this study is like it's very important to work with the bioreactors, because bioreactor give you the platform where you can expand your cells for the transplantation into the humans as well as into the mice. And this scale, bioreactor effectively scale up and differentiate the human iPSC into islets, and this iPSC islets display all kind of islets, hormones, beta cell maturation, which are there. And both stage 5 progenitors and iPSC islets effectively reverse the diabetes, which was the very important factor here, is that the all of this test and all of this work is fruitful because the mice is able to reverse the diabetes. And soon, like in coming days, like there is a human clinical trials is going to start on this, where we are actually recruiting patients to transplant this kind of cells into the patient and see how long they can go for the insulin independence. And apart from this, there is many industrial companies are also there in US who are already working on this, and they have started recruiting patients in this area. So what is unique from the companies who are working, and what is the unique what I'm working right now? The unique part is that this is the autologous study, and it is much more safer for the patient as compared to the allogenic, because in allogenic there is a high risk of having cancers, high risk of forming tumors or having mutation into the cells. At the same time, there's also a possibility that patient has to be on immunosuppressants for their lifetime, and this really breaks the immune system of a patient. And like, let's say, it reduces the life of a patient to 10 years, like staying on the immunosuppressants. So this is the lab. Uh, this is the lab where I've been working. It is a pre-GMP lab, like and we are working also working towards the machine learning and AI aspect also to automate this process of iPSC manufacturing, so that there is less human error. In machine learning and AI, there are algorithms where iPSCs are being trained for the prediction. So this is the machine learning. Next, this is the machine learning overflow, which I'm trying to automate this whole process of this iPSC generation and differentiation using deep learning methods, using machine learning approaches. And these are my next. These are my patents, conference papers. Next, thank you. Thank you everyone over here coming here and listening to my talk. And I'm open for questions.
Right, clamp connection is also for sodium. So it is.
Yeah.
In your previous slide, you are saying that sodium is also involving in clamp connection. So, in my opinion, only calcium will be involved in the clamp connections.
No. I'm sorry to correct you on this. Like
Yeah, sure,
recently, there are some papers in which there is like the presence of this calcium and sodium, both of them, and it is being comparable to the human donor islets. And we did this testing. We did this using different kind of, apart from this patch clamping, because patch clamping is a very superficial. It will just give you the preliminary idea. Okay, this is having or not. But the main thing, like I mostly rely on flow cytometric analysis, I mostly rely on the as I was telling you, gene array, because if someone says like there is a presence of this or not, you can correctly say the answer, I did this gene array and I found this. And in order to publish the papers, like you really have to show that I just did this patch clamping, but there's a presence of this both sodium and calcium of this one.
My next question is, you are saying that ficodilene is there.
Yeah.
Means a
Pilicolin.
molecule. Yeah, pilicolin.
Pilicolin.
A lot of side effect of that molecule is also, I read about that. So
Side effect in what aspect?
In everything, because it is in research that I don't know very much, but some toxicity, some mutagenic effect are there.
No, basically this phylicolin is being used to remove the off-target growth. Basically, the off-target growth is like, let's say I'm going to generate the stem cell-derived islets, and in this stem cell-derived islet, if there is a presence of iPSC, that means this iPSC is a contaminant. That is, this contaminant is off-target growth, and I want to remove this iPSC. In order to remove this iPSCs, I'm using this phylicolin, and I did this toxicology test also, like in-house. The data is not here. In this toxicology test, there's no side effect of this in the mice as well as in the humans.
Okay. My next question is, if you are adding means that molecule, and maybe some carcinogenic or mutagenic would be means we will be grow here, because it generates or it's activate the proliferation of beta cell.
Yes.
So maybe
basically this molecule is being introduced only in the when I'm doing this in a incubators or in a bioreactor. This is not being injected into the mice. So in the mice, when these cells matures, and after maturation, slowly they just remove the off-target growth, and the effect of this phylicolin slowly removes. So there's no possibility of having this. It's not about me only. Like, as I've shown you the pictures, there are many different kind of labs who have worked with this phylicolin at different concentration, and they really didn't find so far.
Okay.
Like maybe in future.
At which concentration you are using that molecule?
The concentration, I'm sorry, this is something.
Okay, understand.
Yeah, I cannot tell you the concentration at this point, but we are using it at a level that it reduces the off-target growth.
Okay, thank you.
Yeah.
So, Ila, just also share that how your PhD work spin off into a company briefly.
Oh, okay. Thank you. Yeah, during my PhD, I have worked with derivation of stem cell, derivation of induced pluripotent stem cells using cord blood and peripheral blood. In this cord blood and peripheral blood, the major limitation is that like in a peripheral blood it's very difficult to reverse the clock. In stem cells are the very basic cells like which are formed when the child comes into the womb. As you were telling, like these are the totipotent cells. Till now, there's no lab in the world who has developed the totipotent cells because of this nature, because they are very much sensitive to maintain. Like they die in like few days. Like no one is able to maintain it for a longer period of time. So I worked with this, started working with this peripheral. Initially, I started with cord blood. I developed the protocol using small molecules, where I started working with this cord blood. Like it took 13 months for me with lots of failures. Like people over here, like with lots of failures in my experimentation. Like nothing worked, sir, for 13 months. I was just hopeless. I was about to quit. I cannot go forward. Like because my boss gave me like you have to generate the iPSCs using small molecules. You don't have to use the viruses. The major limitation of using viruses is at that time they can integrate into the genome of a molecule, genome of a cell, and they might have the tendency to form the tumors. So this is the reason I stick towards the small molecules. And I was working every week, failed experimentation, every week, like just crying behind the doors that nothing is working. So after 13 months, I tried different cocktail of chemicals with different. I did dose dependent study first of all, and at different doses and tested, and fortunately things started working after 13 months. And after 13 months, the protocol initially I developed the protocol, and after development of the protocol, I was able to generate the iPSCs, which I tested using ALP staining. Okay, they are good. Then after I was able to differentiate them into all the three lineages, that is ectoderm, mesoderm, and endoderm. And in this ectoderm, I differentiated into neurons, osteoblast, adipocytes, and lung epithelium. So after I didn't knew that my boss will later on he will start getting into the talks of the patent, because at that time I was about to just finish the PhD and go. No things of doing this patent or spin-off. This happened like I was moving along the way. Like by the end of like my fourth year, I really got very good results. It was very much exciting. The main selling point of my research was that I was using small molecules. I was not using any viruses. I was not using any piggyback cultures which are available in the market. It was just like a small molecules. And this was, and we started speaking for some of the universities in UK, University of Edinburgh, if you know about where the dolly sheep was developed. And we started, and they really got interested, like, okay, this is something very good and we should really proceed forward. So like it took 1.5 years for us to file a patent, and the patent is being filed. And then after, like the university become very much insecure that okay, let's say like if the international student move, what will be the consequences of that. So the knowledge transfer office at the University of Malta came up with the idea that okay, let's go for the spin-off. So there are some, we applied for some initial funds initially, like seed funds for our the company formation. And because the project was very much appealing and interesting, so we got the funds and we started the company. And and since I developed the protocol, and I was the initial person who was there with this project and everything, they decided to make me a director and the board members, one of the board members of the company. Since like I was staying there, and I was having lots of immigration issues. So this is the reason I have to leave Europe and come and started working over here in Canada, because I got my residency status over there. So otherwise, like I didn't have left that company, because I started that. I was one of the founding member of that company. So right now that company is still working, and the main idea behind to get is to develop this reprogramming iPSC small molecule reprogramming kit, which can be, which can be made in a GMP grade environment and can be distributed. Like if you know about stem cell technologies in Canada, they sell that kit. So basic idea about this whole project is to develop the kit to generate the iPSC reprogramming using small molecules in 1 month's time. So this is my journey to get into the company.
It's very great. Yeah, thank you.
Sorry.
You are transferred to the particular lab now, and you are carrying the information with you.
No. I have. They have signed all NDA with me.
So they have already.
No. They have. Then they did the full proof everything with me. Like I have signed. I'm ethically like so. Like right now, sir has asked the question to me, like what is the concentration. So I'm bounded with my ethical thing that I cannot share more information. At the same time, the University of Malta still I'm into the NDA. Like wherever I go, like right now, like next month I'm planning to move. To work on different project in a different place. In that also, like NDA is signed that I'm not able to. I cannot share lots of it. This is the like something which comes up when you are working with the research. So that's it.
NDA students is non-disclosure in
Yeah,
agreement.
it's a non-disclosure agreement, which is which universities or anyone anyone who want to protect their confidential information, they ask other person to sign it.
So, can you name which place you are going to join next?
Yeah, I'm going to join Cincinnati Children's Hospital.
So, can you take some students for internship there from AIMT?
Yeah, sure.
Yeah. So students, please feel free to apply for summer or internship or your 4.5 month internship. Right? You have opportunity to go there and work with Dr. Ela. Isn't it? Okay. So
Thank you.
yeah, Dr. Surbi.
Thank you, ma'am. Thank you for sharing your journey with us, your research, your experiences. I hope our students will feel motivated and they will learn a lot from what you have showed. Thank you once again. Now, I would like to call Dr. Jagriti Tyagi for the felicitation ceremony of Dr. Ela Tiwari Jasra. Jagriti.
Yes.
Thank you. Now, I would like to call Dr. Amit Chandra Kharatwal over the dais and the vote of thanks for the vote of thanks.
So, dear students, this lecture was for you only and to motivate you and see where AIMT alumni works. So, I'm very thankful that Ila could take up time, and come here and deliver a lecture and motivate you, especially so that you also decide to make your career path. Okay, fill the right aspiration survey and do the right progression of your career, right? So it is very important for you to know that there are endless opportunities waiting for you to knock at them, right? So if you decide today what you want to do in a time to come, right, in this day and age, right, of information technology, you can easily achieve your targets. Okay, but you have to work very hard. And I know Ila personally, she worked here in a company for 4 years before moving out for doing PhD. That was all very competitive, right? She wrote many applications, scholarship applications, grant applications, and then finally got one. And then she worked hard there also in the PhD program and then made a spin off and launched a company itself. And now she is also again doing very well and now moving to US in Cincinnati General Hospital, where again she will be working in the area of this, stem cell therapy. That is among the most advanced therapy right now. And I don't think she will be limited to one disease. Yes. Right, she will be working on many diseases. So in future years to come, she will be putting her hands in so many problems, cancer, you name one, there are so many cancers to be worked upon. And this is allogenic therapy is, right, not allogenic, sorry, autologous therapy is going to the future of medical therapy. So she will be known among the pioneers of this therapy, among with other scientists, and I'm very sure that she will take MIT name forward. Thank you very much, Ila. Thank you once again.
Thank you, all faculty members and students, IT team. Thank you so much. Okay, thank you.